We're in the home stretch of the JP Morgan Healthcare Conference. I'm delighted to be joined here in the Flagship studio with Lovisa, who is the CEO of Prologue Medicines and general partner at Flagship. Lovisa, you started thinking earnestly about the company that is today, Prologue, during the pandemic. What was the inspiration for that, and what have you been working on so far since that time?
Great question, and thank you so much for having me here. So yes, we started Prologue during the course of the pandemic because we thought deeply about the viruses, how they affect us, and we were thinking, how can we actually harness the power of the viral proteome and use that as therapeutics? And if we think about the human, we have explored biology for hundreds and hundreds of years, but yet we've only scraped the surface of the underlying wiring of that biology. Viruses, on the other hand, they have existed for millennia, and they have constantly been under that evolutionary pressure to evolve so that they can optimally engage with biology the way that nature actually intended. So that's what we've spent this time on at Prologue, to develop the platform. And that goes both understanding how these viral proteins evolve to have the potency, the specificity, the selectivity to be able to perturb mechanisms in the human body most optimally. But also, if we think about viruses, what they want to do is they want to thrive, they want to survive, they don't want to kill their host, but they want to reproduce. And in doing so, they need to find ways of suppressing the surrounding immune system so that they can thrive, but doing so with low immunogenicity, so that they can still exert their powers. And they've done so by, for example, becoming really small and stable modular protein. So what we've done over the last couple of years is build a platform that allows us to functionally annotate these viral proteins and understand how they engage with human biology.
So you're learning from nature to uncover a new way to target disease.
Exactly.
So let's start there in terms of the initial set of diseases where you think this platform might have its greatest application, and why you think that Prologue is the right fit for some of those diseases.
Yep. So we've actually built out a very broad pipeline over these last couple of years across multiple different disease areas. But where we've chosen to focus initially is really around an immune franchise. We have identified 3 highly differentiated immune assets where we see that they can have both first-in-class as well as best-in-class potential. And these are the 3 assets that we are now are developing through IND-enabling activities so that we can bring them into clinic. And they have each different properties such that we can address different proportions of disease populations within the autoimmune diseases. The first one primarily starting off in RA, the second one primarily starting off in UC, and the third one in CD. And the intention is really to generate early clinical data on these mechanisms and signal search in the clinical population to allow us to then further expand patient populations across also more of those niche, underserved niche indications in autoimmune diseases.
Because as I understand it, a lot of these autoimmune conditions have an underlying shared biology so that you could start in one indication but expand beyond there. And you said RA, rheumatoid arthritis, UC, ulcerative colitis, and what was the third?
CD, Crohn's disease.
Crohn's disease.
Yeah. So these therapeutics have different properties with regards to how they are able to restore immune homeostasis, creating a more balanced immune system with the potential to actually move towards curative approaches. Other aspects in one of the assets is more antifibrotic properties together with the anti-inflammatory properties, and where you can think about in, for example, Crohn's disease, where you see a lot of the fistulas and the types of severe symptoms that will require a lot of antifibrotic properties as well in order to be able to maximally resolve disease.
And are these therapies that you imagine would be given chronically or preventively, or where in the disease progression would they come into play?
So these are primarily, we're starting off in what we call the TNF non-responder population. So those that have started on TNFs and they are not responding in the first instance, or they lose their response to the TNF, either because of anti-drug antibodies or because of other reasons. And we have reason to believe that our first molecule is particularly useful in that patient population. But with what we believe is a very sort of favorable efficacy and safety profile, we of course also look into possibilities of moving from second line up to more first line if you have a safe and tolerable readout in your clinical trials.
Okay, so you have a matured platform, the initial stages of a pipeline that could be quite broad, but a focus area. Tell us about what you're most excited about for the year ahead.
So really bringing these preclinical assets to live and to clinic. I think being able to execute on parallel clinical trials with efficacy or effectiveness, I would say, and being nimble and being able to explore novel ways of running clinical trials, because the benefit here for this asset is that we're really going into known biology, but with novel ways of engaging with that biology. So we can use a lot of the information that is already existing, quantitative medicine frameworks, etc. We know the disease endpoints that we're after. So our goal is to use modern technologies to really identify exactly where we need to collect what data to maximally understand as early as possible what is our differentiation, both from an efficacy and safety perspective, so that our impact on patient lives becomes as great as possible.
So an innovation on the technology side, but also an innovation in the trial design, or how you're thinking about testing these new molecules.
Yeah.
Well, we will be watching with great interest through the year ahead. Lovisa, thank you for joining us here in the studio.
Thank you so much for having me.